So we changed mine a little bit. We're still doing probiotics, we're still doing lactic acid bacteria, but we changed what we're testing. We're going to be doing probiotics versus pathogenic, which we're doing microbial ecology, bacterial competition type test. And what we're testing here is if, like, there's a claim that part of one of the main things that the commensal regular bacteria we have, the reason that probiotics are good for you is because they can prevent pathogenic bacteria infections. And that's what we're testing here, is seeing if they actually do help prevent pathogenic bacteria from being able to colonize. One little twist we're doing from this is most papers I've read, they use pure cultures from the lab. We're doing consumer available probiotics. That way these are like the bacteria that people are actually going to have in their systems, the things that people are actually going to be exposed to. And for this we have, I think it's kefir, I'm not sure how to pronounce it. It's a probiotic milk. We have a yogurt, a buttermilk, and then a probiotic pill. So this is like kind of the game plan. The ones in blue is like what we've done so far, and the black is what's future. So we had to plate our chosen consumer available products, which that's the 2 milks, the yogurt, and the pill. We equally, the pill was one thing we had to dilute it. We had to dilute it in distilled water. And with everything we did, tried to keep it constant, we did 100 microliters of everything onto, we did APT plates. So APT are selective in that for the lactic acid bacteria. That's what we're trying to focus on here. And so we planted them and we grew the bacteria. And then from that, we just did spread plates. We had our lawns of bacteria on our plates. And from that, we started trying to isolate the bacteria, which takes a couple rounds. And after we got it isolated, we were still using APT plates, doing air chambers, so they were anaerobic bacteria to try to mimic what would be in the digestive system, because that would have anaerobic bacteria in there. And then as bacteria, we also, because champagne was just curious, we grew it also on TSA plates and MRS plates, just to see which type of media they grew best on. I don't think I actually did this, I think that was just champagne wanted curiosity there. And then we started doing biochemical tests to try to figure out which bacteria I have. The next step that we'll probably start after a break is purifying the DNA so we can sequence it to see which actual specific species of these bacteria we have growing. And then we'll actually start what the whole thing is about, which is the competition experiment. And depending on how well that goes and how long that takes, we might do some additional testing on like the competition if, like say, if they do prevent growth but there's some pathogenic bacteria still growing on those plates, is that going to affect the amount of lactic acid these bacteria produce? Is it going to affect the pH of the solution? See if it actually makes changes with those pathogenic bacteria there that would affect people's health. Some of my bacteria turned stupid on me last week, and my probiotic pill bacteria just stopped growing. Like it wasn't growing on TSA, which everything grows on TSA, and they just stopped growing, so I had to start over on the probiotic bacteria. And right now I'm back to the plating them, which I did the new lawn spread plates yesterday. So tomorrow hopefully I can start isolating the bacteria to get that, the probiotic pills caught back up so we can get back to the purified DNA where we should be at right now. These are some of my first round of plates I have when we started isolating them. These white looking ones are the TSA. The middle tone ones, those are the APT. And then our dark ones are the MRS auger, which you can ignore those top two, those are the ones that stopped growing on me. But we got 3 from the probiotic pills, we now have the 1 left. The BM, that's the buttermilk. Bacteria we grew from that one. These 2 at the top of the Y, those are my 2 yogurt ones. And there's nothing scientific about how I'm naming them right now. It was just how I could remember which was which. So my Y's are my yogurts, and the C, it was more of a cream colored colonies, and the W is more of white colored colonies. And then my 2 at the bottom are the probiotic milk. And again, same like this, the S is just because they were smoother edges on the colonies, and the R, they were rougher colonies. It was just so I can remember what I was doing right now. And I got my gram stains up here, which, again, we can ignore those 2. The great thing is I'm glad that this third one from the probiotics, it's hard to see on here if you're not like zooming in. But those are actually, they're cocci, they're not bacillus. Which all these, the rod shapes, those are bacillus. That was the only cocci one I got, so I was kind of upset these guys stopped working on me. But hopefully I used the exact same probiotic pill supplied by them. That's my pill supplier for them. And so hopefully we'll find it again. But these are all purple, they're all the gram-negative, I'm sorry, gram-positive rods. Again, it's harder to tell, but my two yogurts, they were also the gram-positive rods or bacilli. Same with, it's a lighter color because part of the stain slightly washed out, but it was still purple. With that milk one, this one over here is my weird guy. We're not sure what's going on with him. He is from the gram stain. I redid the gram stain 5 times to make sure there wasn't some me error making the stains. But this one is actually gram negative. It's pink. We can't quite figure out the shapes. They kind of look rod, at the same time they're not really rods. We're not sure what's going on. And according to all the biochemical tests we did with the different augurs and medias, it should be gram positive. But they're pink with the gram stain with all 5 times I did it, so they're gram negative. And so we're not sure if it's even a bacteria. We think it might be a yeast or something, because it's not behaving how it's supposed to, so we're not sure about that. That's mine and Deb's consensus. Champagne has been doing advising appointments, so I haven't got him about the month. He hasn't looked down yet. But so what this is actually all leading up to will be the competition experiments. That's the whole, our main focus for this. With that, we're just going to be testing once we DNA sequence them and figure out which exact bacteria we have growing. We're going to test them against some pathogenic bacteria, like some Salmonella, some E. coli, some more common ones. And again, we're testing the idea that the benefit of having these probiotic bacteria is that can help prevent the colonization of pathogenic bacteria and infections. So I'll go ahead and start over my bacteria. I haven't got my DNA done yet.
Questions, comments?
Okay, sorry. So, with the, could you go back to the first picture, or no, second picture of your bacteria?
Like the plates?
No, sorry, the gram staining. Yes, okay, so your milk with the rougher edges, okay. So you were saying you think it might be a what?
We're thinking it might be a yeast, because it's not
yeast.
behaving how a bacteria is supposed to.
Yeah.
And from being from the probiotic milk, we're assuming maybe a yeast.
Mm-hmm. Okay, so couldn't you do more, an oxidative test? I can't remember all the tests, because this was like 2 or 3 years ago for me with micro. But could you do like an oxidative test, or?
I did.
You did, and it still wasn't, did it come out like, what was your results for that?
The results came out like they would have for like these, all the gram-positive rods.
Yeah.
That's why like the gram stain 100% does not match in the biochemical test.
Okay.
But we did it 5 times, so like we know, and all the other ones work, so it's not like I don't know how to do gram stain kind of.
Yeah. Of course, Deb would have probably suggested it. I was just like, couldn't you do this and this to figure out? But
I didn't look at it on here, there have been a lot, but I did pretty much every biochemical test we can do in there.
Yeah.
I didn't make them like my MRS. It's like 4 pages of a chart with all the tests, like non stuff I did.
Yeah.
It would take us like 6 slides, and I figured people probably didn't want to care about that. But
Do you think, what was the expiration date? Like, do you think you've gone past the expiration date maybe? Maybe like.
I plated everything the day after I bought it.
Okay.
I think even that milk, I think it doesn't even expire until like December.
Okay.
Like I plated everything right after I bought it.
Because now you got me wondering.
I know. We're not sure what she is.
And was that at a 100 magnification? Or what magnification is that?
Yes, that was the 100 with the oil.
Okay.
I have more pictures. I can see the pictures later.
Oh, I would
More pictures.
love pictures.
Okay, I can.
Love them. Okay.
So how do you plan to do these competition experiments?
That I'm not 100% clear on. You sure I haven't gotten to details of that. We're trying to figure out what we had first, and then we're going to go into more detail on figuring that out. But
So presumably you plate them together in some way, and then one is going to overgrow the other, or?
yeah, There's two ways I was thinking. I'm not sure which would actually scientifically work better. But I was thinking since we already have these probiotic bacteria in us, that we had to have like a plate with like just a solid lawn of these bacteria and then introduce the pathogenic and see if they like kill and take over. And then maybe like mixing like broths of both of them together, plating them at once, and seeing like what takes over.
It's sort of a weird phenomenon, isn't it? That, you know, this whole idea that you can buy this food source and this food source is somehow magically supposed to, you know, I mean, my wife is really into probiotics and all of that sort of stuff, and I'm a skeptic, I guess. I just say, yeah, whatever. As long as it tastes good, it's all right. You know? But I just say that because, you know, I look at animals out in nature and the sort of stuff that they're exposed to. I mean, my dog, for crying out loud, eats bunny poop, eats deer poop, you know, is drinking out of the gutter and all that kind of stuff, and you know that it can't be clean, you know. Other animals eat their own poop, right? So I mean, somehow, you know, this whole notion that you can somehow magically eat something and it's going to make you healthier.
I think there's some things with the feces, it's actually going to help you. I've read some papers about how they're doing like fecal transplants in people that have C. diff, so like trying to rebuild up those good bacteria. So I'm not sure if like the pathogenic stuff just dies faster, so maybe it's not, they're not putting more pathogenic in there. I'm not really sure how that one's working.
So when somebody, you know, when you get a severe case of peristaltic rush, you know, when you've gone down to Mexico and you've had the water or something like that, you've totally cleaned your system out. Have there been any studies to find out how rapidly, you know, that intestinal flora is reestablished?
I'm actually not sure. That'd be a good thing to look into, but I know it really depends on which type of probiotic you're going with. Because like the yogurt, the pills, they're all going to have different amounts, and most of it is getting killed in your stomach with the acid before it makes it there.
Yeah, that's what I would think.
Yeah.
You know?
I'm not sure. I think so. Mostly like I don't know the pills. I think Deb was reading. She bought her drugs in for me, and then there's like a couple. There's like 6 or 8 billion live cultures are supposed to be in there. I'm not sure. It's sitting on the shelf. I'm sure there's thousands of them dying by the minute. I'm sure.
Yeah. The other question I have is just sort of a general microbiology question. I've always had a really difficult time with microbiology just because I don't understand, you know, what the evolutionary relationships are amongst any of the groups. It's easy to figure that stuff out in most plants and animals and so on, but with bacteria, I mean, for example, are all rods in the same clade, or is rod sort of this just secondary feature that can pop up anywhere?
The main big distinguisher is between the gram-positive or gram-negative, and then within gram-positive and negative, you can have the rods or like a little circle oxide.
Yeah.
So it's more like a secondary. Like the biggest thing would be gram-positive or gram-negative, I think.
So shape is something that evolves secondarily, right? And the primary thing is just whether the gram. And what the heck does that mean? I mean, functionally for the organism, what does it mean to be gram-positive or gram-negative?
The gram-positive versus the gram-negative, it's how thick their like outer membrane is, like the peptidoglycan layer. It's how thick it is.
Yeah.
It's gram-negative. I believe it's has a thicker extra layer in there. It's just their outer membrane is the size of the membrane.
Has there been any work on sort of constructing a phylogenetic tree for bacteria?
I think there are phylogenetic trees for them.
Yeah.
I believe.
Okay, so with the gram-positive and gram-negative, it's actually the thinner membrane is the purple.
No, I mix them.
Yeah, you mix them. It's okay. But yeah,
That's
so
weird.
so the positive is the pink and the negative is the purple because the
Purple is positive.
okay, sorry, Mama, look at us. We're gonna each other out.
We're gonna confuse everybody. Yeah.
Yes, so the thinner, because of the thinner membrane, it's going to be purple like the stain is.
Uh-huh.
And because the membrane of the positive is pink, or because it's so thick, it's going to only take a little bit of that stain.
Yeah.
That's why it's pink.
Yeah.
So the actual stain itself is like a dark purple.
Yeah, because the staining, the first one is the crystal violet purple. Then we have iodine to kind of lock it in for the purple ones. Then you do ethanol, that way those thick ones, it'll just wash them right off of it. Then you have the, is it safranin? Safranin? It's a pink counter stain. Safranin, is that? It starts with an S. It's saffron. I think so.
Saffron. Isn't that a spice?
It's like
That's
SAFRS or something.
Are there any other morphological features that can distinguish them apart, or is it all just, DNA?
There's a lot, like, the different, like, sugars they use,
Okay.
the different acids they produce. Which that's what all the different biochemical tests are, like the different
Yeah.
logger plates, the EMB, the DNAs, it shows what they do.
Okay.
And for all of those, they come up, they should all be gram negative to that one. We don't know what she's doing.
Those three are right there, or except for the MR. Y, W, and MS are cocci, right?
No, everything else was a rod besides that one outside. They're short rods, like they're
Okay, I was
short.
gonna say, I was
rod.
like, that one looks a lot. Was the size of the rods different? I'm assuming.
Yes, a lot of these, they have like the shorter, like little stubby rods.
Yeah.
And then these guys have like,
Yeah, like you can
Perfect,
visibly
yeah.
see their rods.
yeah, those two that one down there.
Yeah.
They like had the perfect long ones. But the other ones, once you actually have them zoomed in under the like microscope, the others were like short rods.
Yeah. Did you take measurements of the
Without all
rods?
of that. And then I actually have pictures with the thingy, but I haven't actually like uploaded it on them.
Okay.
Like I have.
Oh yeah, I see the line in there. I see the measurement.
Little measurements on there.
Yeah.
And some of these were even, I think some might be just as kind of like more like the Vibrio, which is like slightly bent rods.
Yeah.
Like,
I was wondering. I was like, I knew there was like cocci rod mix combination. So you think that maybe that might be what those are?
these ones, the Vibrios,
Yeah.
that's what I thought when I was looking at them, but Deb at Quick Glance, she still called them short bacilli,
Okay.
short rods.
Okay.
Champagne will be our defining,
Yeah.
which is this type.
Okay.
Any other questions, comments? Good job. Who's next?
It doesn't matter.
Okay,
Jared talking about how boring and awful bacteria were today.
Right? Like rude.
That's why he
Rude,
brought up the
sir.
bacteria though, because he knew I'm doing bacteria. That's why he's like, oh, or the bacteria. I'm like,
I would have been like, or proteins. They suck too. Click.
or your cells. The cells are not fun.
I don't know if you guys remember my last presentation, but there wasn't much to it. And there's not much to this one either. He's had a busy semester, I guess, Dr. Ruggiero. And he has not talked to me a lot this semester. Frustrating. But I'll be doing stuff with rattlesnake genomes. And we're gonna explore the genome of rattlesnakes he has, like a database that we're gonna be using. He hasn't sent it to me yet. And we'll be looking at transposable elements in the DNA sequences, and then with transposable elements, there's like mutations and insertion points that we'll look at. Okay, yeah, I said that. Basically, like we're gonna be looking at insertion points within the DNA sequences and finding the transposable elements, along with like mutations. The possible projects he mentioned, I think he wants to do the island populations first with the rattlesnakes, and he wants to look at population genetics and how it has affected their like evolution over time, and then like compare the populations and how their genomes have changed, or if they have, and then also look at like transposable elements with the diversity and stuff. And then the Midwest populations, he like briefly mentioned it, like comparing different parts of the U.S. With like their populations, and if they interact with each other like they wouldn't with the island populations, and then comparing those as well. Transposable elements are important for understanding evolution, gene regulation. They increase genetic diversity, which is one of the things that he wants to look at. and then mutations within the DNA sequences. I don't really have much else. He doesn't, yeah, it's not great at communicating.
Who are you working with?
Dr. Ruggiero?
So the, why rattlesnakes? I mean, is it all vipers or specifically rattlesnakes?
I think rattlesnakes, he has done a few papers on them already, and done research with them, written papers, so he just has like more projects that he wants to do with them that he's like already done in the past.
There was a geneticist at Southwest, now Missouri State University, who's now since retired. But he had a graduate, and he was a geneticist, a population geneticist, and his name is Mark McKnight. And he had a graduate student that was working on cottonmouths and copperheads, and he was comparing populations in the Midwest with populations down in Louisiana. And they were doing all of the same sort of transposable elements, genetic sort of stuff. It might be worthwhile sort of getting into the kind of going on ProQuest or whatever and trying to find some of that stuff, because that was it was out of Missouri State, so it was a master's level sort of project. That might be worthwhile exploring, finding out, because I mean, rattlesnakes, I don't know how different genetically rattlesnakes are from, things like cottonmouths and copperheads, but they're all, in the Viperidae, so.
Yes.
You probably don't know, but how, sorry. How would you just like go to like buy the genes from someone? Or are you personally like going to go out to the field and like catch you a couple rattlesnakes?
He, I don't know if it's a paper or a review or a website, but he said that he already has all the data.
Oh.
He just hasn't sent it to me.
Do you know if he's collected it or if
I don't
someone
think
else
so,
has?
no.
Okay.
He doesn't look like the sort of fellow that's out collecting
They just field work.
vipers. You can usually.
He has too many fingers, yeah.
Yeah, he's got all his fingers, so he's
Yeah.
not, yeah.
Mine doesn't, they've never caught anything that's venomous either.
Okay, Sammy.
Do you
Sammy's
care
a
about
resident
rattlesnakes
snake
at all?
chick.
Do you actually care about rattlesnakes at all? Or I'll reword it. Does this actually interest you any, or is it kind of just?
It does, yeah. I'm more interested in evolution and ecology and that sort of thing, which he knows that, and so he's trying to incorporate like population genetics with it and like evolution and stuff. So that like interests me to like put them together.
The reason it's a cool idea is because rattlesnakes are top predators, and they have small population sizes, so population density is low, which means then genetic diversity is going to be spread out pretty far. It's not going to be like mice or cockroaches or something like that. And those populations are highly fragmented, so there might be all kinds of weird stuff going on with, right? And the other thing is that interest in vipers is extreme, especially in the herpetological societies. And it's just like in mammals, there seem to be two types of mammal people: the people that work on bats and then everybody else. And it's the same thing in the herp society. There are the people that work on venomous snakes and then everybody else.
There's the people that just stick to the probiotic good bacteria. People like to do the dangerous stuff with the hazmat suits.
I just think I don't know enough about it yet to.
A really fast question. So Morgan's doing population genetics. Why don't you pick up kind of where she's leaving off?
With her flowers or.
Yeah, or if something else, that, like you can use her work. If you're really that interested in population genetics, why don't you like try to rub off some of her stuff? Because she's gonna leave soon. Not soon, but like you know what I mean, like.
Proposal the other day.
Yeah, she did her proposal and it went really well. I believe it went really well. But it was really interesting stuff. So if you don't want to do the two types of flowers, you can definitely do something else, but like kind of like use some of her work, or you know, because she's doing population genetics, and if that's something that you're really interested in,
Yeah.
skip the rattlesnake, man.
Personally, I just.
Rattlesnakes are awesome. Let's not go dissing rattlesnakes.
All I know is
Andrew was my first.
The other reason for working on rattlesnakes is that there's going to be money available.
Yeah.
Because in Missouri, you have pygmy rattlesnakes, right? You have Massasaugas, you have timber rattlesnakes. So the pygmy snakes and the Massasaugas are I think federally listed. So there's going to be money available for working on those guys. I think you ought to stick with the crotoliths. I think you should, and I think you really ought to go after it because it could be some really cool stuff in there. Good job. Who's next? All right.
So I'm going to be examining the external nasal glands of Eurycia bislineata, also the northern two-line salamander. So my objectives are to describe the morphology of external nasal glands and to determine if they show a seasonal variation or sexual dimorphism. So the glands that I'm specifically looking at are where these two dots are. And the salamander heads are about the size of your pinky nail. So the glands are tinier. But Dr. Siegel was analyzing these heads, and he found these glands just sitting behind the eye, and when he looked at them, he realized that nobody had really gone into the glands before. So the external nasal glands, they were first described in the late 1800s by some German researchers. And then Inez Whipple-Wilder also described them in the early 1900s, but she called them nasolabial glands 1. And then there was a study done in 1932 that compared gland development in different plethodonid salamanders. Some of them showed increased development and some of them did not. Eurycia bislineata showed increased development comparatively to other species. But so far, they've really only studied the tubules. So this is actually a picture from a paper in 1972, no, 75, sorry. They believed that this was the gland, but this is just the tubule. So it actually doesn't show anything, which is why they said that the glands weren't responsible for basically doing anything. They were just like, they just are there. But when Dr. Siegel found them, he found more. When he stained them, he found some more things. So Eurycia bislineata is from the family Plethodonidae, which is a family of lungless salamanders. These ones specifically range from Canada to northern Virginia, and their breeding season is from October to May. So we expect to see possibly an increase in secretions in salamanders that were collected from October to May. Specifically male salamanders, because they're usually the ones that secrete pheromones. So these specimens were collected from February to December of 2017. The reason that none were collected in January because none could be found. So and there usually aren't very many out in January. Both male and female specimens were collected, and about half were fixed in neutral buffered formalin, and the other half were fixed in McDowell's trunk fixative. So the 10% neutral buffered formalin were fixed for paraffin embedding, and the McDowell's trunk fixative were for plastic embedding. So what I'm working on right now is the electron microscopy examination aspect of it. If you look, these are my blocks that I will be that I sectioned, and all of these black dots are glands. It's a lot tinier than you think it is. The sectioning is actually about 1.1 microns thick. But, so I had to dissect the glands, and to do that, I had to peel back the skin of the head of the salamanders, and then place forceps underneath the glands and slowly pull up, because otherwise the gland would just be destroyed. So, once I did that, I went through the process of embedding them in plastic resin, which took about a week for one set of glands. So I did this about 3 times. I section each gland until I find what I'm looking for, and this is about what I'm looking for. So this is a tubule, and all these little tiny dots in it are the secretion. But this is just one tubule in a gland of probably about 15. So I haven't really noticed a whole lot of difference between anything yet, but that's because I haven't really been looking for a lot of differences. I've just been focusing on trying not to lose any glands because it's very easy to do so. And also, Dr. Siegel and I were trying to figure out if something went wrong because some of the tubules were actually staining black for some reason, and we're still not entirely sure why. It could be a problem with the salamanders when they were fixed originally, or the plastic didn't penetrate the tubules well enough. But it mostly just happened with the deep tubules and not really the peripheral tubules. But most glands are actually very usable, so we're just going on. But this is about what I look for, and then once I find that, I have to trim the block so it's just the gland at the tip. So basically, I just have a little tiny thing at the top of my block, and then we'll send them to Indiana, where a man will section them for me, and then Dr. Siegel and I are going to go up there to use their TEM to further analyze what the tubules look like. So then the other aspect of this will be a light microscopy examination. This is basically what it'll look like. These are other glands that are found in the salamander head. So I'll embed them in paraffin. Well, the head's already embedded in paraffin, but I still have to section them for slides and then stain them in hemotoxin and eosin. So this will be about the color scheme that I get. This will pretty much just tell me how big the heads are, or how big the glands are within the head of the salamanders within the different months and the breeding seasons, to see if they actually do show a difference between males and females and across breeding seasons. This would tell us if salamanders actually have more glands that are used in courtship. So we would find out if it differs between seasons and sex of the salamanders, which would further tell us if it possibly could be used in courtship. And then people could further investigate the secretions of the glands to find out if they actually do secrete pheromones that could be used for that. So, yeah.
So the mating season, did I hear you correctly? The mating season is from October to May?
Yeah.
So it's through the winter?
Yeah. They intermittently kind of do it.
So any warm day or whatever, they're going to muster somehow the energy to go through the whole mating. So are these plethodonid salamanders, do the males produce a spermatophore that's deposited on top of the little proteinaceous mound?
I believe so.
So you know where the male has to do this dance, and he
Yeah.
entices the female to put her cloaca right on top of that spermatophore, right? So somehow the male, so the odd thing is, right, it's going to be cold. If you're talking about Canada to Virginia in October through May, a big chunk of that time is butt-biting cold.
Oh, I
So-
know. Yeah.
So these animals, right? They are ectotherms. So somehow,
So I'm from Ohio, and my first salamander that I ever found was Euryssia. It was about 35 degrees outside.
Fahrenheit.
Yeah.
Yeah.
And it had been about 50 the day before, so all of the rocks were nice and warm. But they were plenty out.
Yeah.
So they do like to go outside even though it is a little bit colder outside. I also know that we have a Vistama salamander that likes to breed in about first week of March. It's like key time, even if it's like 20 degrees outside, they're out there. And there's like one good week that you can find them. And if you see eggs, then they're just done.
Yeah.
So,
Now, the fact that some of these glands were totally black on the inside, right? The resin that you're using, I don't know, is this like the resin that you use in woodworking, you know, when you have a piece of hardwood, you're trying to fill the cracks, is that the same kind of resin?
I'm not entirely sure. I've never used that before.
Yeah.
So I'm not sure what the composition is, like how it's similar to each other.
I mean, is it really thick or is it really, really thin and
It's on the thicker side, just while it's stirring. And it takes a really long time to get it up into the pipette to actually transfer it to the blocks.
so it may be that it's just not penetrating into the gland and
Yeah.
what you're seeing there is all this rotted tissue.
So, we have to put it in, it's been so long. We have to put it in, I can't remember what the chemical is, but basically it's what turns the glands black. And so basically all the membranes are just kind of like there, but they're not actually doing anything.
Yeah.
And so if that doesn't penetrate the gland entirely enough, then the resin can't get into it enough. And so that's one of the reasons it could be black. But Dr. Siegel told me that if I had just let it sit in that chemical for a longer time, the gland itself on the outside would have started to disintegrate. And so it wouldn't have been good either way. But we have enough, we've looked through enough slides that we found that we have enough glands to make a conclusion, even if we can't use those particular deep glands.
Yeah, yeah.
So,
What's your success rate between, I know you said you had to pull up the skin slowly or else it would ruin it. And then the resin also had to penetrate it. So,
Freaking rocked it.
if you look at my blocks,
yeah.
you can see some of them have only one and some of them have
Okay,
two.
yeah.
If I was worried about one of those, I would take the other one.
Okay.
So I would usually do that. But so far, it looks like a majority of them are looking pretty decent enough to be able to get some deep tubules. But, you know, just some of them are a little bit touchy.
Yeah.
She did amazing.
Thank you. But, yeah, I have found a lot look like this, and it makes me very excited. But that was my prettiest tubule.
You may not know this, but the picture that you had, just like the random glands, what did the stuff stand for? It was like MG,
NMS and,
CG.
So that's a mucus gland.
Okay.
That's a sebaceous gland, and I forget what the C stands for.
Okay. So I thought MG was something else, but
No.
yeah. Okay.
Magnesium. That's not it. That's
No, that was not what I was thinking. But good, close.
Yeah, I think Dr. Siegel likes to label glands based off of what they secrete, is usually what he does.
So your major prof is Siegel? Why do you have to go to, is it Indiana to do the sectioning?
He has a connection in Indiana that has a
TEM?
Yeah, TEM, and he has, so the ultratone that I'm using is automated, but it's not like good enough, basically, to do what I.
It has to cut through that resin, right?
Yeah, and so for the TEM, you need about 70 nanometers section. And so I can do that with the ultratone that I have, or that Dr. Siegel has, but I would need a diamond knife,
Medical
which is
license?
very expensive, and a steadier hand.
Yeah,
And so, in Indiana, his friend has some special equipment that just, they do it all for you. And so he'll section most of it, put it on some slides for us. And he might have us do some of it, but Dr. Siegel isn't entirely sure. 'Cause
is this at Ball State or where?
I believe it's in Indianapolis.
Yeah,
I believe it's Indiana. It's Indiana State. It's a major. His name is Kevin Burns. I don't know
Yeah,
where
okay.
he works, but I think it's at Indiana State.
we used to have a TEM on campus here. We did.
Yeah. Apparently my old advisor got a TEM, but he's not very good at sectioning things.
Yeah.
So I wasn't gonna ask him.
Good, nice job.